Dear students, today we are going to discuss the topic use of radioisotopes in biology and safety measures in handling radioisotopes. The objectives of the topic are: number 1, to understand what radioisotopes are; number 2, applications of radioisotopes in various fields; number 3, to provide a basic understanding of the principles of radiation safety; number 4, rules and suggestions for the safe usage of radioisotopes and use of radioisotopes in biology. Radioisotopes are atoms that contain an unstable combination of neutrons and protons. The combination can occur naturally, as in radium-226, or by artificially altering the atoms. Atoms containing this unstable combination regain stability by shedding radioactive energy, hence the term radioisotope. The process of shedding the excess radioactive energy is called decay. The radioactive decay process of each type of radioisotope is unique and is measured with the time period called a half-life. Radioisotopes are defined by the Nuclear Regulatory Commission as a new material capable of producing ions or ionizing radiation. The use of radioisotopes in the life science has provided us with new techniques by which we can unravel many of the scientific complexities of the various basic life processes. Now, some of the uses of radioisotopes in biology. Number 1, diagnosis of disease using isotopes. The use of large liquid scintillation counters has shown that they have many practical applications to diagnosis in clinical and experimental medicine. These devices are large enough to assay radioactivity in a whole human body after administration of minute amounts of radioisotope. Measuring such a person for several ensuing days discloses the excretion rates of the isotope from various bodily compartments, and by this means various aspects of human metabolism and disease can be tested and studied. Thyroid uptake of iodine, intestinal absorption of iron, vitamin B12 and fatty acids. The lifespan of red blood corpuscles tagged with radiochromium and absorption and utilization of trace minerals are all among the studies that can be done with these devices. Number 2, medical therapy advances in the treatment of malignancies. With radiations in the past 5 years have developed along the following lines: precise localization of the malignant area to be destroyed by radiation, an ability to deliver an adequate dose of radiation to the site, and a marked reduction of radiation dose to adjacent healthy tissues and vital structures. One important such approach is through the use of the low-energy X-ray emitting isotope palladium-103. This, when injected into tumors as particulate palladium-103 plaque, produces a homogeneous radiation field within the tumor with a small dose to the surrounding tissues. Also, brachytherapy is used to treat malignancies. Number 3, radioisotope scanning techniques. Every drug, once it gains access to the body, tends to follow a pattern of distribution. This localization is a necessary measurement preliminary to any further study. With radioisotope tagging, the distribution of a material inside the body may be determined from outside the body by a method called scanning. Many methods of scanning are already under development: point scanning over one dimension, linear scanning over 2 dimensions, area scanning over 3 dimensions, spectral scanning over the spatial dimension of gamma ray energies, temporal scanning scans the known spatial dimension of time. Combinations of these dimensions or of others include almost every method of detection external to the body. Number 4, nuclear reactor for medical research. The medical research reactor is a heterogeneous tank type reactor operating at power levels up to 3 megawatts. Experimental facilities consist of 2 shielded rooms equipped with special treatment ports and shutters. A broad beam experimental area located at the end of a thermal column is available for whole body irradiation. One of the most promising new techniques of cancer treatment is based on the capture of thermal neutrons by an element such as boron-10. A thermal neutron is captured by boron-10 and ultimately yields lithium-7 and a helium ion plus 2.79 MeV energy in the reaction. The energetic helium ions are easily absorbed by tissue and usually result in the destruction of any living cells in their path. Besides neutron capture therapy, the medical reactor has been successful in producing a number of short-lived radioisotopes useful in clinical research, for example chlorine-38, iodine-132, bromine-82. Number 5. Immunization. Radioactive isotope techniques have made new advances possible by allowing us to label antigens and antibodies and to follow these materials by virtue of their radioactivity within the blood and tissues of the host which receives them, to study rates of antibody formation in relation to time and dose of immunization, and to study the interaction of foreign materials and antibodies within the affected subject as well as in the test tube. Isotope-labeled antigens, once injected into a subject, have been found concentrated in the liver and spleen, often combined with ribonucleoprotein of the cells. Since the ribonucleoprotein probably serves as a template for protein, that is antibody synthesis, this observation suggests that the antigen may directly be involved in the formation of antibody. Isotope-labeled antibodies have been employed in determining the life of antibody in the circulating blood. Number 6. Genetics and cell division. With the advent of radioactive thymidine, one of the principal components of DNA, it became possible for the biologists to label DNA and confirm precisely the earlier finding, namely that once a DNA molecule is formed, it remains intact. This finding demonstrated experimentally that new DNA molecules are built from pre-existing molecules which act as templates to assure the precise organization of the new molecule. Number 7. New radioisotope techniques. The radiochromatographic method is widely used in investigating the chemistry of living things. When radioisotopes are used in the nutrient for a plant or fat to an animal, the locations of the compounds containing the radioisotope are recorded by exposing X-ray film on the filter paper. Radiochromatography has recently revealed the presence and function of two important surfactant components of plants and animal cells. They were discovered as a result of examining radioactive products formed when plants were grown in radiophosphate or in radiosulfate. Of the countless components of a cell, only the radioactive ones are visible on the radiogram. Number 8. Isotope dilution analysis. There are many compounds present in the living organisms that cannot be accurately assayed by conventional means because they are present in such low amounts and in mixtures of similar compounds. Isotope dilution analysis offers a convenient and accurate way of overcoming this problem and avoids the necessity of quantitative isolation. For instance, if the amount of iron in the protein is to be determined, this may be difficult using normal methods, but it can be done if a source of iron is available. This is mixed with the protein and a sample of iron is subsequently isolated, assayed for total iron and the radioactivity determined. Number 9. Production of new plant varieties. Since 1956, a number of new varieties of important crop plants have been released to the farmers as a direct consequence of radiation-induced mutations. These mutations have imparted new desirable characters to these crops which make them more satisfactory than their predecessors. Number 10. Ecological studies. The bulk of radiotracer work is carried out in biochemical, clinical or pharmacological laboratories. Nevertheless, radiotracers are also of use to ecologists. In particular, migratory patterns and behavior patterns of many animals can be monitored using radiotracers. Another ecological application in the examination of food chains where the primary producers can be made radioactive and the path of radioactivity followed throughout the resulting food chain. Number 11. Sterilization of food and equipments. Very strong gamma emitters are now used in the food industry for sterilization of pre-packed foods such as milk and meat. Normally either cobalt or cesium is used, but care has to be taken in some cases to ensure that the food product itself is not affected in any way. Thus, doses often have to be reduced to an extent where sterilization is not complete, but nevertheless food spoilage can be greatly reduced. Cobalt and cesium are also used in sterilization of plastic disposable equipment such as petri dishes and syringes, and in sterilization of drugs that are administered by injection. Coming to safety measures in handling radioisotopes. The radioisotope use is governed by very strict legislation. Work with ionizing radiations is governed by the legal requirements of the Ionizing Radiations Regulation 1999 and the Environmental Permitting Regulations 2010, which supersede the Radioactive Substances Act 1993. The rules are based on the premise that radioactivity is potentially unsafe if handled incorrectly and should therefore only be used if there are no alternatives. Then, once it is decided that there is no alternative, the safest way of carrying out the work needs to be planned. To work safely with radioactive materials, it is necessary to have an understanding of the potential hazards they pose and how to avoid these hazards. Ionizing radiation imparts energy to living cells. A key aspect of determining toxicity is to know how much energy might be absorbed. The higher the energy of the radiation, the greater the potential hazard. The gray and SI unit is the unit used to describe this. 1 gray is an absorption of 1 joule per kg of absorber. The gray is a useful unit, but it still does not adequately describe the hazard to living organisms. This is because different types of radiation are associated with differing degrees of biological hazard. It is therefore necessary to introduce a correction factor which is calculated by comparing the biological effects of any type of radiation with that of X-rays. The unit of absorbed dose which takes into account this weighing factor is the sievert and is known as the equivalent dose. For beta radiation, gray and sievert are the same, but for alpha radiation, 1 gray is 20 sievert. In other words, alpha radiation is 20 times as toxic to humans as X-rays for the same energy absorbed. Clearly, from the point of view of safety, it is advisable to use radioisotopes with low energy wherever possible. Absorbed dose from known sources can be calculated from knowledge of the rate of decay of the source, the energy of radiation, the penetrating power of the radiation, and the distance between the source and the laboratory worker. Dose limits are constantly under review, and although limits are set, it is against internationally agreed guidelines to work up to such a limit. That is, we are not allowed to assume that all is satisfactory if the limit is not exceeded. Instead, the ALARA principle is applied to work always to a dose limit that is as low as reasonably achievable. Work that may cause the worker to exceed 3/10 or 1/10 of the dose limit must be carried out in a controlled area or a supervised area respectively. A potential problem, however, in biosciences is the internal radiation hazard. This is caused by radiation entering the body, for example by inhalation, ingestion, absorption or puncture. There are 3 key principles to radiation safety. Number 1. Time. The less time you spend near a radiation source, the less radiation will interact with your body and consequently your radiation dose will be lower. Number 2. Distance. Distance is a very effective factor in reducing the intensity of radiation incident on the body. The farther away you are from a radiation source, the less radiation will strike you and consequently the lower will be your radiation dose. Number 3. Shielding. In many situations, it is necessary to work with or in close proximity to nuclear materials and or radiation devices. In such cases, shielding is used to stop or attenuate, reduce the level of the radiation. Some of the basic safety measures in handling radioisotopes are: Number 1. Radioactive material signs must be posted on all doors of rooms containing radioisotopes. Refrigerators and cabinets containing radioisotopes must also have a radioactive material sign posted near the opening handle. If at all possible, a specific area in the laboratory should be set aside and clearly posted that it is for radioisotope use only. Second, all bottles, beakers or other containers containing radioisotopes will be labeled with a radioactive material sign or tape, and the following information will be present on the label: type of radioisotope C14, H3, P32, CO60, etc. The amount of radioactivity MCI, CPM, BQ, etc. Date the container was labeled. Number 3. Eating, drinking, smoking, the storage of food and drink in the refrigerators and the use of cosmetics in the radioactive laboratory is specifically prohibited. 4. Laboratory protective clothing such as plastic gloves and lab coats should be worn at all times when handling radioactive materials. This protective clothing shall be left in the radioisotope laboratory. Once gloves have been worn to handle radioactive materials, they should be disposed of in a clearly marked radioactive disposal container. Do not use the same gloves to handle non-radioactive materials, open doors, leaf through notebooks, turn off the light switch, etc. Number 5. While every effort should be taken to prevent radioactive spills and accidents, a small amount of precaution taken prior to an accident can go a long way in minimizing any possible damage. Do not work with radioactive chemicals on the laboratory bench unless they are contained within a spill tray. Number 6. When transferring radioactive liquids, never pipette or perform any similar operation by mouth. Most micropipettes are or can be equipped with automatic tip ejectors. These should be used to remove radioactive tips. Number 7. Modest amounts of shielding can go a long way in protecting the individual user and his or her laboratory co-workers from exposure to radiation. H3 beta particles are extremely weak and cannot penetrate latex gloves. Shielding is especially important if you are working with P32. A beta particle from P32 with its greater penetrating power will travel approximately 20 feet through air. It is more than 99.9% absorbed by a 9 mm plexiglass shield. Number 8. If in the course of work with radioisotopes, personal contamination is suspected, a survey with a suitable beta-gamma surveymeter or wipe tests if working with H3 and C14 must be made immediately, to be followed by the required decontamination. Number 9. When using radioactive material, screw-top tubes are better than flip-top tubes. The more secure the closure of the tube, the less likely it is to pop open, for example during centrifugation or when heating the contents. Securely closed tubes prevent spills. Number 10. Used spin columns can pose a radiation contamination hazard. Place all used spin columns in conical tubes or plastic bags and then into the radioactive waste container to avoid contamination of the lab. Number 11. Ice buckets used to cool tubes of radioactive material frequently become contaminated. Dispose of the used ice in a sink designated for radioactive material disposal. Rinse the sink well to help the ice melt and to decontaminate the sink before anyone else uses it. With this, we come to the conclusion of our lecture. Thanks for watching.